Actually, I admitted all. Actually, nobody in waiting room. 17 members are joined. Nobody is in getting.
In the group, sir, in the WhatsApp group, they are messaging.
Okay, Manikarnika, I'm not able to join the class. Actually, why I'm not sharing the link in the group? Yesterday, so many are joined. Um, ask. Okay. If anyone comes, we'll admit. The delay. Yeah, what is genetics? I think we are discussed in detail like what is the date of birth of genetics yesterday. What is genetics? Rashekar Reddy, what is genetics?
Study of the inheritance patterns of the genes from one generation to another generation.
Yeah, exactly. Anyone? What is inheritance?
Transmission of character.
Can you redefine again?
Transmission of character from parents to offspring.
Yeah, exactly. Transmission of character means horizontal transfer also happens. The transmission of character from parents to the progenies. It is a vertical transfer. What is horizontal transfer of gene? Anyone tell? Can anyone able to tell? It is called. It is not called as genetics. If you are transferring gene from offspring, it's a genetics, like a inheritance. Then the study of that is called as genetics. If the gene was transferring from horizontally without progenies, a gene can also be transferred from one species to other species. It cannot. The study of those things cannot be called as genetics.
Transgenic, we will say.
Transgenic is not a part of genetics.
Gene transfer.
Yeah. Okay. Yeah. Actually, uh, it's redefined. Actually, the whatever the definition you told, it's the very basic definition. Is exactly right. That inheritance, studying the inheritance pattern of the particular gene, then why it is a variation? He told another term called as variation. Like I already it's there in the slide. What is variation?
Sir, difference between two individual uh,
Yeah,
uh, coming from one species.
exactly. Why the variation exists actually in the nature? What's
Like
the main basis?
mutation,
Yeah, mutation. Mutation
mutation,
creates the variation.
uh, hybridization, and
Yeah,
somoclonal.
hybridization, somoclonal variation. Yeah, the many ways the variation can be created. The study of the variation and how that variation are transferring from one generation to other generation or across the species or anything maybe. That's why genetics again redefined as the science that deals with structure, science deal with structure, organization, transmission, and function of the genes and their origin and also the variation present in them. Okay, this is what exactly the proper definition of a genetics. Who given the term genetics? Who given the term genetics?
Bateson.
Yeah, Bateson. Which year?
1903.
Not fine.
Yeah, exactly. 1900, 1901 afterwards, 1901, 1903, 1902, whatever it may be. Um, Bateson actually given the term genetics. Who given the term? Genetics is also having the term, another term called as gene. Who given the term gene?
Johansson.
Yeah, Johansson given the term gene. Then I have an announced quiz for you all. Please comment, please respond your answer in the comment box, or what is you say, response box. What is gene? I will give you 2 minutes, like 1.5 minute time, 1 minute or 2 minute time. Please write the definition of a gene in the comment box. Everyone, I will check everyone. Okay. By the time I will see what's up. Please write quickly. I am already late today. Sorry for that inconvenience. Actually, nobody is in the waiting room. It's very shy. You're writing chat box. Please write need definition. Okay? Don't text like a WhatsApp text, WhatsApp chat text. Yeah, I think I have received the charts. Oh God, thank you for telling. Thank you, Sumit. Jageshwar, fundamental unit of heredity. Yeah, yeah, okay. I think I got response from. That's good response. Sumit, the answer. Okay, Jageshwar, why you defined a gene as a fundamental unit of heredity?
Sir, actually, I said functional unit. Sir, mistakenly
Yeah,
fundamental.
no, there is no mistake. Why okay, what is why it is functional unit of heredity?
Sir, gene actually Gene is a sequence of DNA
Yeah.
which code any polypeptide
Yeah.
जो functional unit है
Yeah.
actually
Then you're saying that gene is a sequence of DNA,
DNA sequence
DNA sequence, a DGC.
which codes for a functional product of polypeptide या ऐसे के mRNA
mRNA.
RNA
Yeah, RNA. Okay, whatever it means. Not code for mRNA. RNA. Any RNA. Okay.
any RNA.
Then if it is a promoter, it having a promoter, it cannot be called as a gene because promoter not code for RNA. Cannot be called as promoter, terminator, enhancer, activator sequence, some UTR sequences. This cannot be called as gene. Yeah, we'll come to the next your definition. Gene is a functional again heredity. Gene is a unit. Rashekar Reddy. Gene is a unit of having the cistron, recon, muton. Why you have said this definition? Can you be able to explain? Because I don't know this definition. Can you explain me?
No, sir. Actually, gene is the functional unit of heredity. By means of the cistron is the responsible for the passing function of the genetic information, and recon part is responsible for the recombination, and mutant.
Sorry. Can you start again? I am unable to.
Gene is having the 3 units of the cistron, recon, and mutation, sir. Each part is having the different function, like mutant having the mutation function, recon having recombination function, and cistron is the functional for the genetic information. Altogether leads to the passing of the information from one generation to
What is the why it's defined as if, for example, if you're saying this cannot be, so you are saying that there is a gene, there are 3 different parts. One part is coding for a gene, another part is for crossing over, another part is for mutation. Is it like that?
Yes.
Sure.
All genes not like this, sir.
No. I'm okay. Whatever the definition you told, it's exactly right. Okay, as of now, the explanation you given is you're having a confusion.
Okay.
You have confusion in the way of the explanation. Like I'm asking that gene having a 3 different parts. One part is giving a product, another part is specifically for recombination, another part is specifically for mutation, as you were saying. I'm right?
Yes.
Okay. You are sure about your definition? Then we'll
Yes,
go
sir.
to another definition. Please, I think it's there on record. Yeah. Gene is actually mRNA sequence that codes for a protein. Saumajit Chakrabarti. Gene is a mRNA sequence. Then what is the sequence of DNA? Cannot be called as gene?
So actually, it's a coded form. Means for expression,
Okay.
it needs.
Gene having what is split gene concept? No, you know what is split gene concept?
Yes, sir.
Can you explain?
Split gene means both having intron as well as exons.
Yeah, intron cannot code for mRNA. Then it cannot be called as gene. Yeah, I think I will come to you. Then again, functional unit of heredity. Yeah, fundamental and structural unit of heredity. What is the meaning of fundamental? And why you're saying again structural unit of heredity? What exactly it means? What is fundamental and structural? What you
Sir,
want to convey?
functional and structural, I wrote because sir, gene means it has specific DNA sequence, so that and it has as well as it has specific function. So we can tell it has a structure as well as it has a function. So that sir, I wrote like that.
Fundamental and structural unit of heredity. Okay.
Yes, sir.
All the definition are right. I'm not saying that any definition is wrong, but I'm asking explanation. I want to know functional unit which governs. See how many definitions are raised. There is a only one gene. Like sorry, many genes are there, but the gene is same. But answers and see the how much variability exists here as like the variability exists among the genes. Functional unit of heredity, functional structural unit of heredity. Functional unit which governs the development of many traits. If any the gene which is not governed for any trait, then it cannot be called as gene. See, for example, the genes coding for siRNAs and miRNAs, they will not even convert it into the protein or expression. Expression part is protein level. If the gene is not even expressing in the form of proteins, then it cannot be called as gene. Shubhamji Verma?
Yes.
No, I'm asking question. It cannot be called as gene.
What, sir?
Like you have answered, gene is a functional unit which governs the development of many traits. I'm asking
Yes.
that some genes will code for only miRNA, siRNA, transactive acting RNA. Okay, let it be anything. Which not able to convey. It's not able to convert into a protein. Then it can't. Particular sequence cannot be called as just gene.
Sir, maybe no, sir. Sir, because it development of various characters of individual is necessary for the gene, sir.
Yeah. Okay, we're right. Okay, but I will come to you again. Then Venkat, gene is a sequence of DNA that starts that exactly gives one functional polypeptide or one mRNA molecule that may also include intron as well as exon. Can you heard about? Can you heard about gene within a gene? Can be any term gene within a gene? Fintron.
Yes, sir. Also,
Yeah.
if alternative splicing can happen, and there are many mechanisms, there are overlapping genes, etc. sir. So,
Actually, noise
two
is coming.
genes,
Okay. Your
two
voice
genes
is
can
like
be, sir.
yeah.
Now I am
Next
clear, sir.
can now okay somewhat clear. What is nested gene?
Sir, a gene which is completely within another gene, sir. It may be within
You are
an.
saying that a sequence of a DNA coding only one RNA. If the sequence have also a coding for another RNA, cannot be called as gene.
Sir, actually, the starting and the end point of a gene can vary, sir. Two genes can overlap within each sequence, sir.
Yeah, I'm asking. I'm asking. Okay. Yeah, you are partially right. Gene is a structural unit of a living organism. Virus cannot have a genes. Virus is living or non-living?
Non-living.
Yeah, non-living. Then how it is replicates, propagates? It's a semibiotic. Okay. Sometimes it acts as a non-living, sometimes it will act as a living. Means gene, we can't say gene present in virus also. Maybe it is present in semibiotic organisms also. Okay, we can't say that only living organism. Generally, it's a living organism. The basic unit of heredity passing from parents to offspring. Okay, only the parent, only adulthood. If gene is transferring from one species to other species, without passing to the progeny cannot be called as gene again? Yes. As you all are right, actually. That is why gene, the concept of gene is not a static. It doesn't have a single definition. As you answered so many answers, it is called as concept of gene concept. It's a very dynamic. We will see how exactly you said it's a, but no one said Mendel's definition. I kept a beautiful animation of Mendel. Can anyone tell what Mendel has said about the gene? Actually, my lectures are like this only. Like it's a more interactive than putting everything in the slide and reading the slides. Like if I read the slides, if I kept every information, no difference of reading the book by yourself, reading by myself. Please answer.
This
The
is good,
definition
sir.
of the
Factors.
factor. What is the factor? How Mendel has imagined the gene. How Mendel has described the gene. Yeah, each factor covering a trait, as he said, as one of the guys said. Actually, it's a debatable question now. Somebody saying if the ultimate point is trait, I will come to that later. Mendel said any factor, it's a factor which codes or governs a particular trait. Mendel concept of gene. But today, but at Mendel, when he given this concept, 1866. But when the genetic material was discovered, like the proper structure of genetic material were discovered. Can anyone tell?
19.
When DNA is proved as a genetic material, can anyone at least?
Avery McLeod experiment, sir.
Yeah, I am asking when.
1928, sir.
1928. I'm asking when DNA is proved as a genetic material. You said 1928 is wrong. 19?
Hersey and Chase.
No, I'm asking when. Santoshini,
Sir,
you
in
joined?
1950.
So I think 1920.
See, one guy
Griffith
answered
X-ray.
experiment name. I'm asking the date. See, the basic things you have forgotten.
1920, sir. I
Yeah,
think.
exactly. Jageshwar, 1944. 1944 is the exact answer. You should remember the basic things were there now. These are very critical in the point of examination. They ask even, see, in my Viva, like in my final Viva of my MSc, while actually I was explaining the DNA replication, semi-conservative model of replication, the examiner asked like, why how many number of cycles he grown in the N15 media. Can anyone tell? Okay, let it be, we'll discuss later. Like that, you should be a very, very point to point, you should be very short when it comes to the basic fundamental milestones. 1944, actually the DNA is proved as a genetic material. See, the Mendel don't know even the genetic material, but he defined something that there is a factor which governing for a particular trait. It is actually the basic definition. And he also said the trait is inheriting from one generation to next generation by his monohybrid and dihybrid trihybrid crosses experiments in the nearly 20 plus species of the any plants. Then, yeah, discuss. What there are two definitions. Like it's not a classical definition generally is a unit of function, a recombination, mutation. I will come to later why it is exactly a unit of function, recombination, mutation. But for the time being, we will see what is a modern definition of a gene. It's a sequence of DNA, and it is not perfect. I'm saying, like how disclaimer. You can take it as a disclaimer. This is not perfect. This definition is not perfect, but by the time being, we'll see. Sequence of DNA that specifies one polypeptide or one M molecule. This unit includes a 5' upstream and 3' downstream, a non-coding regulatory sequence, as well as entrance of a gene. As when Venkat has responded, it exactly a right answer, but it is not perfect answer. There are so many objections. Can you guess what are the objection? Can you think any objection can be raised by looking at the modern definition of a gene? What I pasted here.
Sir, one
Think of this.
polypeptide.
Yeah, that can be one. Yeah, another.
Even one RNA also, sir.
Yeah, okay. One mRNA, one polypeptide. Okay, that can also be. Then any other? The one concept is there. You know, if any antigen comes to your body, there will be n number of antibodies. N number of antibodies. And it's generated instantly. How it can be generated instantly? See, if antigen A comes, it will produce A. Antigen B comes, it will antibodies B will produce. C comes, C will be produced. D comes, D will be produced. How instantly the gene able to the system can able to produce this number of antibodies with a short number of time?
Pleiotropies.
This concept, please. This is an assignment. In the next class, please tell that answer. Okay? This concept,
Pleiotropy, sir.
not the pleiotropy. Pleiotropy is another thing.
Sir, many mRNA can be produced.
Yeah, that can be, but another thing is there. Please answer in the next class. This is assignment. Please search this. If you search this, you will remember for the lifetime. This is one objection. I will tell. Okay, if I tell you the surprise, the suspense will be lost. I told you the concept already. If the antigen comes to the body, like the system can produce a number of antibiotics within a short duration of time. That mechanism is itself an objection for this definition. Please answer in the next class. Next, I will explain how the gene concept has evolved. Like as you said, there is so many definitions. Exactly, each and every definition is correct, but it's having an evolution from one era to the next era. See, in the 1866, the Mendel said it's a unit factor. It controls a specific trait. For example, you take plant height or a color, whatever it may be, it's governed by one gene responsible for one character. Then, later in the 1902, after the rediscovery of the Mendel, Garrett, you know what is the famous book was written by Garrett? Can anyone tell?
Inborn errors of metabolism.
Exactly right. Inborn error of metabolism. When he is studying the alkaptonuria disease, it's an autosomal recessive disease. I hope you are very clear with what is autosomal, what is allosomal, what is sex linked, sex limited, sex influenced. I think many of you know what is the difference between them. It is a autosomal recessive gene, the controlled by autosomal recessive gene. While studying this trait, alkaptonuria disease, he concluded that one mutant can lead to govern the one metabolic block. Here, one gene, one trait. He narrowed down, for example, you take it as a long, it's a trait. He narrowed down this much. It's a metabolic block. What is metabolism? What is a metabolic block? What exactly it's defined as? Can anyone respond? Abhishek, Panavada, Abhishek. Am I right? The spelling. Santoshini.
Catabolism plus anabolism.
What is catabolism and what is anabolism? See, I'm telling If you are defining something, you should be precise and you should be in a one shot. You should clear. You should be able to convey the what you are saying. Right? As you said, catabolism and anabolism. Then question arises: what is catabolism? What is anabolism?
A separate pathway for growth.
Exactly. It's in a simple way. Any pathway having so many enzymes, so many genes, any pathways means the total metabolic path. For example, in the case of Garrett, here he actually there will be a sorry I wrote made a notes about because even I also forgetting the spellings. Actually, this alkaleptonuria is a disease. It's caused by the lack of one enzyme, homogenesic acid oxidase. I'm poor in spelling. I'm saying by homogenesic acid enzyme. If this enzyme is not present, what happens? There is no conversion of homogenetic acid into methyl acetate acetic acid. You read in the books, I can't write here. Means if there is no change, if there is the metabolism is disrupted, like there is a conversion of homogenetic acid to the methyl acetoacetic acid. If this metabolism is disrupted due to there is a change in there may be a change in the one gene. Not proved actually. He said that there may be a change in the gene can leads to disruption of the metabolism of the alkaptonuria. So there is an accumulation of this compound in the urea when it is came exposure to the air become black. I think many of you know that story. This why he defined like okay something is the any change in the gene can disrupt the metabolic clock. But Mendel said if any change in the gene directly a trait, from trait to he narrowed down to metabolic block. Are you get? Have you got my point? It's understood for everyone? Any doubts in
sir.
this? Then another development has happened. See the time gap. See the time gap after 40 years. The Beadle and Tatum, genius scientists, this told that one gene specifies one enzyme. See, in a metabolism, there are so many enzymes. Let's ultimate product. I think many of you know that, like what is this one gene affect another gene, then another gene, then another gene. Finally, we'll get some product. This is the entire metabolism. As per the Garrett, entire metabolism governed by a single gene. As per the Mendel, the whole trait governed by a single gene. As per the Beadle and Tatum, each gene is governed by a separate genes A, B, C, D, E like this. Earlier, whole it is as a. Then some split metabolism, A metabolism, B metabolism, then leads to particular trait. But it is narrowed down in 1940, it's still narrowed down to, okay, there are several different genes that coded by, there are several different enzymes in the particular pathway or in the particular metabolism that can be governed by a single gene. Have you got my point? I'm not going in detail because many of you read many times. This is called as one gene, one enzyme hypothesis. He conducted experiment in the Neurospora and its mutants. What is that experiment? Can anyone tell? The experiment conducted by Beadle and Tatum. I am not dealing those things because it's a very like basic things you are learning from 7th standard. I think. What is that experiment? What Beadle and Tatum is conducted? Can anyone tell?
They treated
You should
Neurospora
explain.
spores with X-rays.
No. You should explain in a 5 to 6 steps. First step, second step, third step, fourth step, fifth step, like that. First step, what he did?
He treated with a mutation.
How? For what? With what is treated? Why is treated?
He treated with UV rays, yes, sir. UV rays, I
Yeah,
think, sir.
no.
UV rays.
Okay, you are telling that Neurospora fungi is treated with a UV rays. Then what happens?
Yes.
Yeah.
Then the mutants are formed from the spore.
Yeah, then second step.
The mutant is lacking in one of its function, creating a specific enzyme. So
How we came
before
to
it,
know? See, you are directly
Before
jumping
he-
to conclusion. I told
Okay, sir.
that steps. Experiment. How you conducted the experiment? You directly told the conclusion.
Neurospora
Next,
will grow
after
in
creating
limited
growing
media, sir, ma'am.
minimal
That is
media.
minimal media.
Minimal, yeah, minimal
Media.
media. Okay, then third,
Then the mutants are unable to grow in minimal media. They are growing in complete media only.
like okay, then fourth,
Then he provided each media separately. He divided
each
the
media
vitamins. He divided the carbohydrates like that.
yeah, okay.
Then he identified that its growth is limited by the vitamins. So he provided different vitamins separately.
Yeah.
Then he identified that vitamin pyridoxine, or biotin, something like that. He identified that because of its limitation, it's unable to grow in
Yeah,
the minimal
exactly.
media.
As you said, yeah, exactly. What you said is 100% right. First, he created the mutants. Then, either he compared with wild type or not.
Yes, sir.
You
He
mentioned
compared.
that point.
He crossed it with wild type like that, sir.
Yeah. Then he crossed with. No, he did. Why? If we cross, what will happen?
After crossing only the spores he obtained. He treated those spores, sir. After crossing, he treated those
Yeah.
spores, I think.
Okay. Generally, you say it. First, he created the mutants so that each mutant having some dysfunction. He grown those mutants separately in separate minimal media as he did, and each media is deficient in several amino acids. For example, you take is a proline, some it is a or any amino acid like guanine, methionine, something. If, for example, the particular fungi cannot able to grow in the minimal media which is lack in the proline, that mutation may be responsible for the biosynthesis of proline. Means that mutation directly involved in the enzyme which coding for the proline biosynthesis. By that way, he concluded that each mutation governing a particular enzyme. Okay? Now you understood, everyone understood what he explained. It's exactly right. Anybody got anyone anyone had doubt in this experiment? Please, everyone, please respond. Any doubts?
No.
Now I think you understood from a trait to metabolism to an enzyme, how the evolution of a gene concept taken place. Then another enzyme is what is enzyme? What is enzyme? Forget about the spelling. Just write in the slides. What is enzyme?
Combination of polypeptide, protein starch, and then metabolic structure.
Okay, combination of polypeptide that takes place in the metabolism. If particular enzyme it is not involved in any metabolism, cannot be called as enzyme.
Or some enzymes are involved in feeding the metabolic blockchain.
Simple way, enzyme is just a sequence of polypeptides. Then it's like again is every polypeptide cannot be an enzyme. This is another exception. But enzyme it's having a different polypeptides. Different polypeptides. And each polypeptide may be governed by different genes. That can be possible. That's stated by Ingram. One gene, one polypeptide. The famous experiment, like at first time, not he only studied. By this experiment, like sickle cell anemia. Can anyone tell what is sickle cell anemia? Next time. What is sickle cell anemia?
Sir,
These
due
are
to
the
mutation in hemoglobin, hemoglobin gene, some mutation in hemoglobin gene,
no.
the red
You
blood
should
cells will form sickle cell shape.
see what I am asked. You tell some. What is it? Some only I asked. I wrote already hemoglobin. The some mutation means you have to specify. What is sickle cell anemia? Exactly you define, like in a precise way.
Red blood cell sickle shaped.
I already wrote sickle cell anemia itself having the name sickle cell. See, actually, normally we 1 minute. Actually, normally we we are suffering with the one disease. I don't know that name. Something like some like for example, you take bioinformatic analysis or these things. If we don't know that, we'll feel like insecure that okay, that things I don't know, these things I don't know. By in that process, we forgetting the basics. Like the many students fail in the SRF examination due to this fear actually. That's why read the basic. If you are very good in the basics, you can easily crack SRF or ASRB net. ASRB net is a very easy exam. Like if you're very clear with the basics, you can able to clear. Okay, we'll come back to the thing. Please define again. You are right actually. Please define again. We'll try at least. What is sickle cell anemia? Starting 3-4 classes, I will be dealing like this only. After that, I will be in a very good speed. What is sickle cell anemia?
Loss of blood due to change in polypeptide sequence from glutamine to valine.
Exactly, glutamine to valine. And a mutation which causes the change of amino acid glutamine to the valine, by which the change in the nucleotide, which mutation causes that?
Beta
In which
chain,
position of the amino acid?
sixth,
Beta chain is okay.
sixth position.
Yeah, exactly. Sixth position. At what transition? That mutation I'm asking. Like which nucleotide mutation will cause the sickle cell anemia? A to B transition mutation. Is a transition mutation or transversion mutation? Can anyone tell? Adenine to thymine. It leads to
It's
change of
transversion, sir.
glutamine to adenine to thymine. This transition or transversion?
Transversion, sir.
Yeah, exactly. Glutamine to valine due to this A22 transversion mutation, or like I'm telling transition, like when there is a is replaced with a T, there will be a change in glutamine to valine at the sixth position of the, as I said, beta chain of hemoglobin. The sickle cell will occur. Actually, you should remember, you should behave it, because in every exam, it will be going to ask, because it's a first disease is studied at molecular level. Then see the hemoglobin having 4 polypeptide, 2 alpha and 2 beta, and it is each polypeptide governed by A and B gene. Like sorry, say 2 different genes. Means Beadle and Tatum said each enzyme governed by a single gene, but here each polypeptide is governed by a different gene. So that the concept of one enzyme, one hypothesis is gone. It's not gone. It's one of the exception, because so many enzymes are monopeptides. If the enzyme is polypeptide, its enzyme is constituted by polypeptides, then the one enzyme, one gene, one enzyme hypothesis is failed. Okay? These are how the gene concept is evolved by the time being with the invention of new things. Then the C. Yanofsky, first time while studying like he deciphered the sequence of tryptophan synthase A protein. While doing this, he proved the hypothesis which provided by Crick. The hypothesis is called as gene protein collinearity. Means there is a sequence in the nucleotides ATGC. It will exactly collineate with the sequence of amino acids in the protein. If there any change in the nucleotides of the DNA, there will lead to change in the amino acid. This is called as gene protein collinearity. Understood? Any doubts in this? What is the exception for this?
Degeneracy of
genes.
genetic code, sir. Several codons can promote to same amino acid, sir.
Yeah, that can be done. That can be one exception. Any other exception? Like twintron or nested genes. Gene like nested gene means gene within a gene. They are the exception for this collinearity things. But as of their time and as of to some things, those definitions are fitting. But there is always an exception. Okay? There is always exception for everything in the biology. That's why in CSR normally there is a one trick. Like if it is only the thing that is always this thing, is if you catch that word, you should put the statement as false many times. Okay, this is one trick. Then we move to the next. Sorry. This sorry. Actually, this is a different. Open the different slides. This is nice side. I made modifications, but okay. Actually, the paper was published by Bidley and Tatum in the 1941, October 8. Please remember this date also. Sometimes maybe it will go to us. And which journal he published, that also important. I forgotten actually, I have taken the screenshot. See, the many of the invention came from the US. Yeah, I wrote. Then, what is this? What is this picture? This is the different types of mRNAs. Like, sorry, I'm not RNAs. tRNA, mRNA. See, there is also an exception because so many the RNAs will not code for a protein. So this is another exception for this. Then, yeah, I want to say this. Even now, as we see that the concept of gene from 1866 to 1957, 1960s, it is actually the conference happened in, is the date, 2017. It's but entire conference about the conference is happened in Japan or Germany. Sorry, Germany. They can't able to define exact definition of a gene. Please read this paper. Why till now nobody cannot able to define what exactly the gene it is. Please read this paper. The evolving definition of the term gene. It's published in April 1, 2017. Please read this. It's a very important. It's published in the journal Genetics. It's a very like a reputed journal in the genetics. Please read this paper for this. Okay. Then I will come to the definition told by Rashekar Reddy. Can you repeat your definition again? What is the gene, like structural, functional, some unit you told? Can you redefine? Can
Yes.
you again? Yeah.
Gene is having the three parts, that is cistron, recon, and mutant. Cistron is responsible
Yeah.
for the functional, recon for the recombination, and mutant for the mutation.
Yeah, okay. See, earlier, the gene can be defined as a beats on string concept. See, these are the beats which are on the string. Means this is one gene, this is one gene, this is one gene, this is one gene. And there are different alleles. A allele, small allele, for this particular gene, this particular gene, this particular gene. As it said, gene is a structural, is having a structural unit, functional unit, and unit of mutation. Means what exactly it means? A crossing over cannot able to like what we can say, indivisible, like it divisible, cannot able to divide the gene. Crossing over cannot able means gene you can't split. If you split this gene, the functionality of the particular gene will be lost. Have you understood my point? For example, it is a one gene. If you make it to two different parts, or if you divide this gene, it can able to do any function. Means, for example, if crossing over happens between E and F, this crossing over cannot split the gene, or the crossing over cannot takes place within the gene. Understood my point? Means a gene within a gene you can't able to recombine. Means it is the structural unit. Understood what is exactly means? For example, if what is cell? What is cell? What is cell?
Structural and functional unit of an organism.
Structure. Why it is defined as structural and functional unit? Means if you break the cell into 2-3 parts, you can't able to do function can be lost. Understood? Cell can be divided. Okay, cell if you break the cell structure, the function will be lost. Means it is the smallest unit. If you break this one, the function of the particular unit will going to last. That's why when we define fundamental, structural and unit, fundamental structure means if we break this structure, the function governed by the structure will be lost. This is actually what exactly means fundamental, structural, functional unit. What you will say the definition of it. Cell. The same way gene means this particular unit, if you divide or if you do anything, the function of that particular unit will be lost. That's why it's a basic fundamental structure which cannot be which is individual by the crossing over, and if there is any changes in this particular gene, there will be a mutation will happen. If there is any change in this one, there will be a mutation happens. Then another one is it is governing for particular proteins or a functional product. That's why yeah, please tell.
Sir, but there is also an evidence for intragenic recombination.
Okay. I will come to that. I'm about to tell. Okay. I'm about to tell that. Then this is called as a beats on string concept. Means the genes are look like a beats. This beats cannot be divisible by crossing over. If any change in this beat, it leads to another allele. It is called as mutation. Then the particular gene having some function which codes for a protein. This is actually a classical definition of a gene. As he said, there is an intragenic recombination. Intragenic recombination. Who given this term? Intragenic recombination?
Oliver, sir. Losante Lopez.
Yeah, Oliver. That leads to the another thing like structure of a gene. Functional allelism and structural allelism. we will come to and we will discuss, I think, in tomorrow's class by the complementation and recombination test. Read complementation test, recombination test. Maybe I will not go into detail because it's not that much important. But you read how the intragenic recombination happens and how we proved that within a gene there will be a recombination. How we, based on that, there will be two terms: structural allelism and functionally allelism. If gene, if for example, if two mutations, if you want to say that they are functionally allelic and structurally allelic, what are the criteria need to be satisfied? Please read this concept. Tomorrow, I think we will discuss. Okay. Any doubts until now? This is actually beads and string concept. But after that, after 1940, Oliver and Benzer experiments has disproven this. Now, the modern definition of the gene came based on those experiments. I will deal tomorrow. Okay. I think I have crossed the time, 1 hour. Then any doubts? Any doubts regarding the concepts till now? Any doubts? Please ask. See, as I am repeating again, this is the only chance we can able to interact. Okay, because many times I am not able to reply properly in the WhatsApp text. As of this is the time I am giving, like 1 hour. It's a very important time. You have to discuss more, learn more.
Sir,
Even I also learn more.
so recombination test is of entirely useless, I think, sir, according to
What? Can anyone tell?
recombination.
Your voice, you're having very noise actually. Like so much noise is coming along with your audio.
Hello, sir. Now I'm audible, sir.
Yeah, now you are clearly audible.
Sir, a recombination test is entirely became useless after this intragenic recombination, sir.
No.
Yes, sir. That was my.
Tomorrow we will discuss. Okay. Structural allelism of particular mutants was exactly given by the recombination test. If recombination test itself failed, how you map a gene?
Yes, sir. That was my doubt, sir. Okay.
Tomorrow, please read this chapter from the book. Which book? You know, any book you have read? Yeah, exactly. Nested and Simmons. It's a very good book. He explained in detail. Please read this chapter, and tomorrow I will deal with it, if possible.
Okay.
Because I want to start Mendelian genetics and molecular genetics a little bit according to schedule, but I want to discuss these things because even in my MSc first class they discuss these things only. So just to get a curiosity, I discuss these things. Okay. Any doubts up to this? Okay. What are the things I will going to discuss tomorrow? Don't ask that. Up to this, any doubts? Like anything? Okay, please, if you're liking if you like the class, if you feel interested, please share among your friends so that they can also get benefit of the classes. Okay. Please give the feedback. I received only 2-3 feedbacks. I think many of the, if you not give the feedback, it may regarded that you're not liking the classes actually. Because based on your feedback, I will prepare the lectures for the next classes. I will improve so that it will benefit you and me also. Thank you everyone. If you have any doubts, please ask now or you can ask in the text.
Sir, is all the classes regarding the molecular breeding will be complete, sir? Or it will be such an conceptualized thing, or it will be complete class, sir? I'm asking that.
No, it will be in detail. How much I know, I'll how much I know the concept, like how much I understood, I will tell everything. Okay, it's not like that. I will keep something as hide and something I will. It's not like that. Because if I explain more, I will also learn more and it is easy to remember, and it is to write as a like answer in different exams for me also.
Yes, sir.
Okay? That's why I'm highlighting. Even I will also make the notes before coming to class. See, that's why I'm very sure, Shah. This is why even you also, like many people think like spoon feeding, like they expect spoon feeding. It's not like that. Even I also, if I want to teach, before like half an hour or before 1 hour, I will open the book, I will make the notes, I will revise it, then I will speak. Without reading, it is very difficult to remember. I know, like some acids I had to remember, some enzyme I had to remember. It's very difficult. That's why you should be you should be stick with the books. You open the first one. Some tricks I need to tell. First, you download the syllabus, paste on your walls, like any room or anywhere. You paste the syllabus on the walls. Then every day, whatever you read, you just stick. For example, the first line of syllabus is, I think, the Mendelian principles. I think general genetics. In the unit of general genetics, there is a first line is Mendelian genetics. Write that word Mendelian genetics, and you should read all the things which are related to Mendelian genetics and uh, tick that word in the syllabus. Then you shift to next. You should only focus on two things if you are preparing for any exam. One is syllabus, word by word. You should know at least the meaning of each word in the syllabus. Then how much you read and the notes you are making. Whatever you read, you make the notes. If you just simply read the book and you'll forget, okay? That's why first you choose the topic from the syllabus, you make the notes out of it, and you revise. Then only you will be able to clear any concept, either it's easy concept. See, even this is a basic concept. Even I am in PhD second year. Okay. Although I am reading and I am making the notes now also. Because why? Because when you read more, when you write more, you will uh, perfect more actually. You will become more perfect. Okay. If anything is there, please ask. Any doubts? Please share your friends so that if number increases, it will be interesting to me to teach also. Please share among your friends, and if you're liking the classes, please give the feedback so that I can improve. Thank you. Thank you, everyone. If any doubts, you can text. Shall I end the class?
Yes, sir.
Thank you. Thank you.
Thank you, sir.